Usually when we talk about a medical diagnosis, there's this, expectation of precision.
Right, like engineering.
Yeah, exactly. You
Yeah.
break your arm, the x-ray shows that jagged white line, and the doctor just points at it and says, well, there it is.
It's very binary. It's broken or it's not broken. And I mean, it's comforting because we really like things to be visible and easily categorized.
We do. But then you step into the world of neurodevelopment, and suddenly that x-ray machine is practically useless. We're looking at a diagnostic landscape that is, well, it's murky.
Very murky.
Welcome to the deep dive. Today we are looking at the USMLE roadmap for neuroscience to figure out exactly how the nervous system wires itself together, and more importantly, the bizarre, terrifying things that happen when those wires get crossed.
Which is an incredibly ambitious journey for you to take with us today.
Mm-hmm.
We're going to explore the underlying logic of the brain and the spinal cord. So not just memorizing anatomical names, but really understanding the mechanical why and how behind neurological function. That way you can actually apply this in a clinical setting or, just understand your own biology a bit better.
Okay, let's unpack this. Because if you think about the sheer scale of the human nervous system, we are talking about billions of neurons, trillions of connections. Where does the body even begin building a network like that?
Well, it all starts super early in the third week of embryonic life.
Yeah.
And it begins with this tiny, simple structure called a neural plate.
Just a plate.
Just a little microscopic plate. But from that beginning, it develops into a system so staggeringly complex that 40%...
Like 40?
Yeah, 40% of the central nervous system's neurons end up being dedicated just to processing vision.
40%. Just to see. That's wild.
Just to see. It is a phenomenal architectural feat. But before we can really understand the complex wiring of the adult brain, we have to look at the raw materials, right?
The building blocks.
Exactly. So if we go back to that third week of embryonic life, the embryo undergoes a process called gastrulation.
I remember the term from biology, but what is gastrulation actually doing mechanically?
Essentially, it's the embryo sorting its cells into three distinct layers. You've got the endoderm on the inside, the mesoderm in the middle, and the ectoderm on the outside.
Okay, inside, middle, outside. Got it.
Right, and within that middle layer, the mesoderm, a rod-like structure, forms. And this is called the notochord.
The notochord.
Yeah, and the notochord starts sending out chemical signaling molecules, like retinoic acid.
So it's basically like texting the cells around it, telling them what to become.
That's actually a great way to think about it. The notochord sends these chemical texts up to the overlying ectoderm layer, commanding it to thicken.
And that's the neural plate we talked about.
You got it. That thickened area is the neural plate. So as the cells multiply, that plate folds inward to create a groove. And eventually, the top edges of that groove pinch together, creating a closed tube.
Oh.
We call this the neural tube.
Gotcha. And what does that tube eventually turn into?
So the neural tube is a foundational structure for the entire central nervous system. So your brain and your spinal cord.
Okay, so that handles the central part.
Right. But during that pinching off process, a few cells get left behind on the outside of the tube. These are the neural crest cells.
And they just kind of hang out there.
Well, because they are on the outside, they wander off to form the peripheral nervous system. So that includes the sensory nerves in your skin, your autonomic ganglia, things like that.
Okay, so we have a hollow tube for the central system and these wandering cells for the peripheral system. But what about the cells themselves? Because a neuron doesn't look like, a normal round skin cell or a flat muscle cell.
Oh, far from it. Neurons are highly, highly functionally polarized. They have a receiving end and a shipping end.
Like a warehouse.
Exactly. The receiving end features these tree-like branches called dendrites, which pick up signals. The signal travels into the main cell body, which houses the nucleus, and a massive amount of rough endoplasmic reticulum.
Why so much rough ER? That's the part of the cell that manufactures proteins, right?
Yes, exactly. In neurons, we actually call it Nissl substance. Neurons are incredibly active. They constantly need new proteins, enzymes, and neurotransmitters to function.
Makes sense.
But here is the logistical nightmare for the cell. The cell body makes all these proteins, but the shipping end, the synapse where those chemicals are actually released, can be up to a meter away.
A meter?
Yeah, all the way down at the end of a long, wire-like structure called the axon.
A meter away. So if a cell body is in, my lower back, its axon could stretch all the way down my leg to my big toe.
Yep.
How does the cell get the supplies all the way down there? It's like having a factory in Chicago, but the retail store's in Miami. You'd need some sort of cellular freight train system.
That analogy is remarkably accurate, actually.
Yeah.
The axon has microscopic tracts called microtubules.
Okay.
So to move cargo from the cell body down to the terminal, the neuron uses an enterograde transport system. It attaches the cargo to a motor protein called kinesin. Right. And kinesin literally walks down the microtubule tract.
Just walks down it. That's amazing. But what if the terminal has, I don't know, empty containers or chemical feedback that needs to go back up to the factory?
Then it uses the retrograde transport system. So a
Yeah.
different motor protein called dynin carries recycled materials back up the tracks to the cell body.
Man, this sounds incredibly efficient until you realize that some nasty pathogens have figured out how to use this freight train for free rides. The reading mentioned the rabies virus. How does a virus exploit this?
What's fascinating here is that the mechanism is just terrifyingly brilliant. When an infected animal, like a skunk or a bat, bites you, the rabies virus enters your peripheral muscle tissue.
Right.
But it doesn't just multiply in the blood, it gets taken up by the local axon terminals in your muscle. It then attaches itself to that dynin motor protein.
And rides the retrograde train all the way up the axon into the central nervous system.
Exactly. It's actively hijacking the inward-bound train to get into the brain, completely bypassing normal immune defenses.
That is just, wow.
And it gets worse. Once it reaches the brain and replicates, it jumps onto the anterograde consin train, riding it down the nerves that lead to the salivary glands.
Packing the saliva full of the virus just in time to make the host aggressive and bite someone else. That is dark.
It's very efficient, biologically speaking.
Yeah, I guess so. But let's assume the freight trains are carrying their normal cargo, not rabies. When those signals reach the end of the axon, they trigger the release of neurotransmitters.
The chemical messengers.
Yeah.
The two most critical ones to understand here are glutamate and GABA.
Glutamate and GABA.
Okay,
Yeah.
so glutamate is your primary excitatory neurotransmitter. When it crosses the synapse and binds to the next neuron, it opens sodium channels, causing the cell to fire. It basically yells, go.
And GABA.
GABA is the primary inhibitory neurotransmitter. It opens chloride channels instead, making the next neuron negatively charged and much, much harder to fire.
So it's like the nervous system's main set of brakes yelling, stop he. If GABA is the brakes, how do we use that in medicine? Like, can we force the brakes down if someone is having a seizure or extreme anxiety?
That is exactly the mechanism of action for benzodiazepines.
Oh, like diazepam.
Right, commonly known as Valium. Diazepam doesn't replace GABA. It actually binds to the GABA receptor and makes it far more sensitive to the GABA that's already there.
So it essentially presses harder on the brakes, calming the electrical storm in the brain.
Exactly. Okay, so we've looked at the single cells, the freight trains, and the chemical messengers. Let's zoom out a bit and look at how these millions of neurons bundle together to form the spinal cord.
The superhighway.
Yeah. The anatomy of the spinal cord is fascinating because it's actually the inverse of the brain. If you slice a spinal cord in half, you'll see a white, fatty-looking outer layer and a gray, butterfly-shaped inner core.
Why the color difference? I mean, why is the outside white?
The white matter is on the outside because it consists of the myelinated axons. These are the tracks carrying signals up and down.
And myelin is the insulation.
Right, myelin is a fatty insulation, which naturally makes it look white. The gray matter on the inside contains the actual cell bodies and the synapses.
And I noticed the text mentioned that the gray matter is organized into these layers called rexed laminae. But, functionally, how is this butterfly shape split up?
It's pretty straightforward functionally. The back half, the dorsal horn, is entirely dedicated to receiving sensory input from the body.
Okay, dorsal is sensory.
And the front half, the ventral horn, is entirely dedicated to sending motor output to the muscles.
Ventral is motor. Okay, let's follow that motor output. If I decide to move my hand, how does the signal get from my brain all the way to my muscle? Because the text mentioned upper motor neurons and lower motor neurons, and I always see medical students get tangled up on this.
It's a classic stumbling block. Think of it as a two-part relay race.
Okay.
The upper motor neuron has its cell body way up in the cortex of the brain. It sends a long axon all the way down the white matter of the spinal cord, but it never actually touches a muscle.
It doesn't.
No. It simply delivers the command to the lower motor neuron.
So the lower motor neuron takes the baton.
Yes. The lower motor neuron lives in that ventral horn of the gray matter we just talked about. It receives the signal, sends its axon out of the spinal cord, and physically plugs into the muscle fiber to make it contract.
Got it. So the upper is like the general in the command tent, and the lower is the soldier out in the field. How does a doctor tell which one is damaged just by examining a patient?
By looking at the reflexes. It's really interesting. If you cut the upper motor neuron, say, from a stroke in the brain, the muscle becomes weak, but the reflexes become wildly hyperactive. We call this spastic paresis.
Wait, why would they be hyperactive if the nerve is cut?
Because the brain normally sends inhibitory signals down the upper motor neuron to tell the spinal cord reflexes to calm down.
Oh, I see.
Yeah, if the general is gone, the lower motor neuron soldiers just go rogue. Every little tap on a tendon causes a massive, unsuppressed reflex.
Wow, but what if you cut the lower motor neuron instead?
Then you get flaccid paralysis. The actual wire to the muscle is severed. The muscle goes completely limp, reflexes disappear entirely, and the muscle quickly atrophies because it's no longer receiving any electrical stimulation to stay alive.
Okay, that makes perfect sense. So that covers the motor commands going down. How does sensory information get back up to the brain?
We have two main sensory pathways, and they are strictly segregated.
Okay, what's the first one?
First is the dorsal column medial lemniscal system. This tract travels up the very back of the spinal cord and carries fine touch, vibration, and conscious proprioception.
Proprioception, that's your brain's awareness of where your limbs are in space, right?
Exactly. Knowing where your arm is without looking at it.
And then the second pathway.
The anterolateral, or spinothalamic system. This tract travels up the front side area and carries pain and temperature sensation.
This brings up a massive clinical puzzle. I want to throw a scenario at you based on the text.
Sure, go for it.
Let's say a patient suffers a spinal cord hemisection, meaning exactly half of their spinal cord is severed by some trauma, let's say on the right side. Why on earth do they lose motor function on their right side but lose pain sensation on their left side?
It all comes down to the precise anatomy of where these microscopic wires cross over from one side of the body to the other.
Okay.
Let's trace the motor pathway first. The upper motor neurons coming from the brain cross over very early, way up in the brainstem, long before they travel down the spinal cord.
But the wires for the right arm are already on the right side of the spinal cord.
Yes. If you cut the right half of the spinal cord, you are cutting motor wires that have already crossed. Therefore, you lose motor function on the same side as the injury, the right.
Makes sense.
The dorsal columns for fine touch do the exact same thing. They travel up the right side and don't cross until they reach the brainstem. Cut the right cord, lose touch on the right.
But the pain pathway has a different route.
The spinothalamic tract fibers for pain are the rebels. They enter the spinal cord and immediately cross over to the opposite side within 1 or 2 segments and then travel up.
Oh, wow.
So the pain fibers traveling up the right side of the spinal cord actually originated from the left side of the body.
Sever the right spinal cord and you lose pain sensation from the left side.
Exactly.
Mm.
This mismatch deficit is the hallmark of Brown-Séquard syndrome.
It's incredible how a tiny difference in where a tract decussates, where it crosses, completely changes the clinical presentation. And speaking of pain, the body doesn't just passively transmit it, does it?
Not at all. The spinal cord actively processes it. There's a gate control mechanism in the dorsal horn that can block pain signals if activated by other non-painful touch.
Like rubbing your elbow after you're bumping.
Exactly. Furthermore, the brain can send signals down from areas like the periaqueductal gray in the midbrain to actually suppress pain at the spinal level. Your body has its own built-in analgesic system.
That's fascinating. Let's follow those tracks upward into the skull. The spinal cord thickens and becomes the brainstem. That's the medulla, the pons, and the midbrain. This area is tiny, but honestly, arguably the most critical real estate in the body, right?
Without question. I mean, the brainstem controls your vital autonomic functions like breathing and heart rate. It also houses the cranial nerves that control your face, your eyes, and your throat.
And because everything is so densely packed together in the brainstem-like, the cranial nerves going out to the face and the long motor tracts passing through to the body, a tiny stroke here causes a very specific pattern called a crossed sign.
Yes. A cross sign is the defining feature of a brainstem lesion. It means a patient presents with a cranial nerve deficit on one side of their head, but body weakness on the opposite side.
Let's visualize that. Walk me through the mechanism of a cross sign in the lower brainstem, like the medulla.
Okay, imagine a stroke damages the medial or inner part of the right medulla. The hypoglossal nerve cranial nerve 12, which controls the tongue exits right there. So the right side of the tongue becomes weak.
So when they stick it out, it deviates to the right.
Correct. But what about their arms and legs?
Right, what happens there?
That same stroke damages the corticospinal tract, the upper motor neurons for the body, before they have crossed over to the left side of the spinal cord. As a result, the signal to the left side of the body is cut. The final presentation is right-sided tongue weakness, but left-sided body weakness. The geometry perfectly pinpoints the lesion.
It's like a map. While the brainstem keeps the heart beating and the face moving, we also have two massive structures attached to it that dictate how we move: the cerebellum and the basal ganglia. I used to think these structures directly force muscles to contract, but they don't, do they?
No, they are the upper management. They don't touch the lower motor neurons at all. They manipulate the motor cortex in the brain, refining the commands before they are ever sent down the spinal cord.
How do their jobs differ? Like, what does the cerebellum do versus the basal ganglia?
Think of the cerebellum as your quality control manager. It operates on a feedback loop. When the brain decides to move your arm, it sends a copy of the blueprint to the cerebellum.
Okay.
As your arm actually moves, proprioceptive sensors in your joints send real-time data back up to the cerebellum.
So if I reach for a coffee cup and my hand is slightly off target.
The cerebellum compares the blueprint to the actual movement, calculates the error, and instantly fires corrective signals back up to the motor cortex to adjust your trajectory mid-reach.
That's happening instantly.
Yes. And if the cerebellum is damaged, you get ataxia or intention tremors. The arm shakes wildly as it gets closer to the cup because there's no error correction.
Wow. And if the cerebellum is quality control, what are the basal ganglia?
The initiators. They function like a complex circuit of gas pedals and brakes to allow desired movements and suppress unwanted ones. You have the direct pathway, which ultimately excites the thalamus, giving the green light to the motor cortex.
Okay, and the indirect pathway.
That pathway puts the brakes on the thalamus. When you want to move your arm, the direct pathway says go. But simultaneously, the indirect pathway tells all the competing muscles in your body to stay quiet.
So this balance ensures smooth motion.
Exactly.
Where does dopamine fit into this machinery? Because we always hear about it with Parkinson's disease.
Dopamine comes from a structure called the substantia nigra, and it acts as the ultimate optimizer. It turns up the direct pathway's gas pedal and turns down the indirect pathway's brakes.
So it just makes everything easier.
Right. It makes movement easy to initiate. In Parkinson's disease, those dopamine-producing cells die off. The brakes clamp down, and patients have a terribly hard time just initiating a step.
Here's where it gets really interesting, though. The basal ganglia aren't just for physical movement, are they? Because the text outlines a cognitive loop.
This is one of the most fascinating aspects of neuroanatomy. The prefrontal cortex projects into a parallel circuit in the basal ganglia. But instead of initiating a physical movement, this pathway initiates executive behaviors. It organizes verbal skills, problem solving, and coordinates socially appropriate responses.
So the brain uses the exact same gas and brake circuit architecture for physical movement as it does for filtering social behavior.
Yes.
So if you have a lesion there, you might see impulsive behavior or a loss of social filters simply because the neurological brakes on inappropriate actions are failing.
Precisely. The mechanism is the same, only the context changes. And that leads us to the highest levels of the central nervous system, the diencephalon and the cerebral cortex.
Okay, let's talk about the gencephalon. It sits right in the center of the brain. It's got the thalamus, which we mentioned is like the ultimate relay station. Almost all sensory information going to the cortex has to stop at the thalamus first.
Right.
And below it is the hypothalamus. The book uses the acronym HEAL for it, right? Homeostasis, endocrine, autonomic, and limbic.
Yes. When we say homeostasis, we mean it acts as the master thermostat for your internal environment, regulating temperature, hunger, and thirst. The limbic system, on the other hand, is the primitive emotional brain.
The mnemonic for the limbic system here is home, homeostasis, olfaction for smell, memory, and emotions. I want to drill down on memory, though. How does the limbic system turn a passing thought into a permanent memory?
It relies heavily on a loop called the Papez circuit.
The Papez circuit.
Right. This connects the hippocampus to the mammillary bodies of the hypothalamus and up to the thalamus. This circuit acts as the processing plant for memory consolidation, turning short-term experiences into long-term storage.
And when this processing plant gets damaged, the results are pretty devastating. The text specifically highlights Korsakoff's syndrome.
We typically see Korsakoff's in cases of severe alcoholism due to profound thiamine deficiency. Thiamine is crucial for cellular energy, and without it, the mammillary bodies degenerate. The Papez circuit basically breaks.
And these patients develop severe anterograde amnesia, like they physically cannot form new memories. But the bizarre part is what their brain does to compensate.
They confabulate. Because their brain cannot accept the gaps in time, it invents highly detailed, wildly untrue stories to fill in the blanks.
And they believe it, right?
Completely. And the tragedy is they genuinely believe these fictions. It's not intentional lying, it's a mechanical failure of the memory circuit.
That's heartbreaking. So, from the deep centers, the signals finally reach the wrinkled surface of the brain, the cerebral cortex. This is where we have highly specific, mapped-out functional regions.
Often referred to by their Brodmann area numbers.
Right.
For instance, area 4 in the frontal lobe is your primary motor cortex, dictating voluntary movement. But clinically, the language centers are paramount. In the dominant hemisphere, which is usually the left, you have Broca's area, Brodmann areas 44 and 45.
And Broca's area handles the actual motor production of speech. If a patient has a stroke here, they suffer an expressive, non-fluent aphasia.
Mechanically, what happens is their language comprehension is perfectly intact. They know exactly what they want to say, but the motor plan to move the lips, tongue, and vocal cords cannot be assembled.
So they can only force out broken, frustrating syllables.
Exactly. It's very frustrating for them. But Wernicke's area, which is further back in the temporal lobe, handles the comprehension of language.
Area 22.
Right. A lesion in Wernicke's produces a receptive, fluent aphasia. The mechanical production of speech works perfectly, so they speak effortlessly.
But because comprehension is destroyed, the words come out as just a word salad. It lacks any meaning. And the real tragedy here is that, unlike Broca's patients, a Wernicke's patient generally has no idea they aren't making sense. Their comprehension center is broken, so they literally cannot comprehend their own deficit. They often get angry at the doctor for not understanding their perfectly fluent nonsense.
If we connect this to the bigger picture, we must remember these areas don't function in isolation. Broca's and Wernicke's are physically connected by a thick bundle of white matter axons called the arcuate fasciculus.
What happens if a stroke hits that connecting wire but leaves the two centers intact?
You get a conduction aphasia. The patient can comprehend you perfectly, and they can produce fluent speech. But because the two areas can't communicate, they absolutely cannot repeat a sentence back to you. The wire transferring the information is cut.
The brain is so entirely reliant on these white matter connections. The cortex communicates down to the spinal cord via the internal capsule, which bundles millions of motor wires into a tiny space. A millimeter-wide stroke there can paralyze an entire half of the body.
It's a huge bottleneck.
What about sensory processing in the cortex?
The parietal lobe integrates sensory information. A fascinating example of localized function here is Gerstmann syndrome, caused by a lesion in the inferior parietal lobule.
When that sensory integration fails, it's not just numbness, right? It's much weirder than that.
Yes. Patients develop a calculia. They lose the conceptual ability to do simple math. They develop finger agnosia, completely unable to recognize or name their own fingers.
Wow.
And they lose the spatial awareness to tell right from left. It highlights just how compartmentalized higher cortical functions are.
Let's finish our cortical tour with the most real estate-heavy sense, the visual system. We talked about it dominating 40% of the brain earlier. So the light hits the rods and cones in the retina, gets converted into electricity, travels down the optic nerve, crosses at the optic chiasm, and hits our favorite relay station, the thalamus.
Specifically, the lateral geniculate nucleus of the thalamus. From there, it radiates all the way back to the primary visual cortex in the occipital lobe at the very back of the skull.
But arriving at the visual cortex isn't the end of the line, is it?
No, the brain then splits the image processing into two distinct streams. You have the dorsal wear pathway, which projects upward into the parietal lobe. This stream mechanically processes motion and spatial awareness, so you don't, walk into walls.
And the second stream.
The ventral what pathway, that projects downward into the temporal lobe and handles the high-resolution processing of object recognition, color, and distinguishing faces.
Wow. We have covered an immense amount of ground today. We've traced the nervous system from a tiny embryonic neural plate down the freight trains of the axon, navigated the complex crossings of the spinal cord and brainstem, balanced the gas and brake pedals of the basal ganglia, and finally mapped out the calculated conscious cortex.
It's a dense amount of material, but understanding these structural pathways is the only way to accurately localize lesions in a patient. Let's actually test what you've learned.
Oh, a pop quiz for the listener. I like it.
Exactly. Imagine a 59-year-old patient who have a right homonymous hemianopsia, meaning they can't see the right halves of their visual field in either eye. However, when you shine a light in their eyes, their pupillary light reflex is perfectly normal. Where the lesion? Take a second to think.
Right visual field gone, but light reflex works.
Right, yeah. Because the pupillary light reflex is fully intact, the sensory limb going through the optic nerve and optic tracts to the midbrain is unharmed. the lesion must be further back, downstream of that reflex arc. Therefore, relying on the anatomy, the lesion is likely in the left primary visual cortex or the left optic radiations.
Localizing the lesion based on pathway logic, not just guessing. This is why we care about the mechanism. So what does this all mean for you? We started by talking about how the nervous system isn't like a simple X-ray of a broken bone. It's a complex, dynamic web. We just spent all this time rigidly mapping out the cortex, like this is Broca's area, this is Wernicke's area, this piece of tissue controls the thumb.
Right.
But if the map is so incredibly rigid, how does the brain's capacity for neuroplasticity allow some patients to completely recover speech or motor function after a massive stroke destroys those exact areas? If the structural X-ray is broken, how on earth does the wiring fix itself? It's something to ponder as you continue your medical studies. Thanks for taking this deep dive with us.